All right, that was step 5. So let's go ahead and do step 6. So we're going to make sure that we have talked about what's going on in each of these phases. We're going to really focus on the words homologous chromosomes and sister chromatids. So let's go ahead and finish this off. All right, so we already did prophase 1. That's where that crossing over happens. So let's go ahead and talk about metaphase 1. So this is where the homologous pairs line up in the middle of the cell. So that's one thing that's going on here. You'll also notice that we got these things right here. Those are the spindle fibers. So I'm going to go ahead and label that for myself. And they are what attach to the centromeres and kind of act as like tethers to eventually like pull these chromosomes. So let's go ahead and make a note of that. So spindle fibers attach to chromosomes. Okay, so that's metaphase. Keep in mind that chromosomes can like trade places, so they don't have to have like maternal side versus paternal. They can flip-flop. All right, so they lined up in the middle. Now we're in anaphase. Notice what's happening here. We know these chromosomes are being split apart. But what's different from mitosis is in anaphase 1, it's the homologous pairs that get pulled apart. So this is kind of a really important thing here. And this is kind of where diploid cells are going to eventually become haploid because we are now going from 6 chromosomes to 3 on each side. So homologous pairs are pulled apart. Okay, so that's a big thing that happens here. This ensures that we are going to, here's our sheet again, make sure that gametes contain half the number of chromosomes. That's because of what happened in metaphase 1. All right, or anaphase 1, sorry. Okay, so after anaphase 1, we have telophase, cytokinesis. What's happening here is this cell is dividing into 2 cells, which we can see right here, okay? So after telophase 1 and cytokinesis 1, we now are at prophase 2. And we're gonna hit pause here for a second. Notice, we do not go through interphase ever again. We only go through interphase at the very beginning. So once we get to cytokinesis 1, we jump right to prophase 2. There is no interphase. Remember, interphase is making copies of DNA. We already cut this DNA into half. Like, we now have 3 chromosomes each. We don't want to double it back up again, okay? So that's why interphase does not happen ever again. It only happens once and at the very beginning. So prophase 2, the spindle fibers forms again. The cell is prepared to divide for a second time. Okay, so that's what we're seeing here. Metaphase 2, notice the chromosomes. They're lining up in the middle. So let's go ahead and write down chromosomes. Line up again in the middle of the cell. Okay, remember these 2 big chromosomes used to be homologous pairs. We pulled them apart in anaphase 1. Okay, so this pair is in this cell, and this homologous chromosome is now in this cell. We separated them. All right, metaphase 2, chromosomes are lined up in the middle. See these spindle fibers, they're attached again. We now enter anaphase 2. What's going on here? Why is this different from this? In anaphase 1, it's the homologous pairs that are pulled apart. Anaphase 2, it's the sister chromatids are pulled apart. Okay, so these two sisters used to be connected. Okay, they used to be connected by a centromere, but that chromosome was pulled apart. So we now have this sister on this side and then this sister on this side. So they've been pulled apart. Remember, sister chromatids, this is still considered a chromosome. This is still considered a chromosome. We've just pulled them apart. All right, so after anaphase 2, we now go into telophase 2 in cytokinesis, and this is what we end up with. Okay, so we have 2 cells here, 2 cells here. So what happened in telophase 2 in cytokinesis? The cells pull apart, and that's cytokinesis, resulting in 2 cells. So 2 cells here, 2 cells here. So we now end up with 1, 2, 3, 4. We have 4 cells. These are gametes. So describe the end product. We have 4 genetically different, keyword here, gametes. Okay, so let's pretend these were sperm cells. So what would eventually happen is these cells would undergo something called spermatogenesis, if they're sperm, and they would grow tails. Okay, so I'm just going to draw them here on mine. If you want to make yours egg cells, you can. But egg cells have a weird end story. So I will be talking about that in a bonus video. So yes, I'm going to make a 4th video here. So you guys are lucky. All right, so here we have 4 sperm cells, genetically different, because if you really look, notice we got 1 large blue chromosome here. On this side, it contains blue and pink. This one over here contains blue and pink, and then this one's a completely pink one. Okay, so that is where the genetically different gametes come from. And again, have we met our goal? Have we made gametes that contain half the number of chromosomes? Yes. Are they all genetically unique? Yes. All right, so that is meiosis. Thanks for watching.